morning, viewer. Welcome to our Glory Globe TV as a breaking news. In a new study published in Cell, researchers describe a newfound mechanisms for creating proteins in a giant DNA virus comparable to a mechanism in eukaryotic cells. The finding challenges the dogma that viruses lack protein synthesis machinery and blurs the line between cellular life and viruses. Protein production is accomplished in cellular life by decoding messenger RNA, that is called the mRNA sequences, in a process referred to as translation. In fact, most genes have some function related to protein synthesis, however, viruses are not and do not contain cells. In contrast to living organisms, viruses cannot replicate independently and rely on a host cell to perform many of the biological processes required to reproduce. Although viruses encode proteins involved in DNA replication and transcription too, the dogma is that all viruses share a universal dependence on the host cell translations machinery for viral protein synthesis, explained the author of a new study. Giant DNA viruses are a little different. These viruses, which can be as large as bacteria, possess unusually large genomes and encode genes with homology to cellular translation factors. They typically infect unicellular eukaryotic hosts, most notably amoeba, algae, and other protists. Previous research showed giant viruses encode tRNAs, that is called amino acyl tRNA synthetases and putative translation factors. However, researchers were still uncertain why giant viruses encode these translation factors and whether they are functional or evolutionary remnants too. Now, the team involved in the new study set out to determine whether the giant DNA viruses possess a functional translation initiation complex and how it might impact viral replication. As a model, they used the giant virus Acanthamoebia polyphaga mimivirus, that is called APMV, or mimivirus 2. They used mass spectrometry, crystallography, and homology searches to identify proteins, which they refer to as viral IF4A and viral IF4E. So the proteins and homologous to the proteins ELIFA and ELIF4E found in eukaryotic cells. The team found that the proteins come from a functional cap binding translation initiation complex termed B1F4F in the virus, which is homologous to eukaryotic UIF4F. So here we discovered that giant DNA viruses encode an IF4F mRNA can binding complex characteristics of eukaryotic protein synthesis and that controls translations during infections too. Viral IF4F coordinates synthesis of structural proteins during late stages of infection, and this transcript specific regulations occurs through evolutionary innovations in the viral IF4F mediated M74 cap recognition, the study author explained. Then the exclusive report.